Okay, so we're going to talk about cell communication still. Previously, we did an overview on what cell communication is, the reception, the transduction, and then the response. What we're going to do now is focus just on the reception and the types of receptor molecules that are present or found in cells as a way of facilitating communication. So let's do a quick overview of cell signaling once again. We have reception, so the signal has to be received. That's what we're going to focus on in this video. Then we have to have transduction, where we're going to relay signal throughout the cytoplasm until we ultimately get our response as some sort of activation. Usually we're making or stopping making a protein. Okay, so reception. When signal molecule binds to any receptor protein that is located in the cell membrane, it's going to cause it to change its shape. Because remember, proteins are fluid molecules. They can have conformational changes. Usually what happens is we have to think of the proteins in the cell membrane as being similar to like enzymes in a way. And the reason is because they are very specific for shape. So shape determines function. So it turns out that the binding between a signal molecule, which vocabulary-wise we call a ligand, and the receptor molecule that's in the cell membrane is highly specific. And so there's actually different types of receptors for cell communication, and then there's unique receptors for every ligand or signal molecule that is going around our body. Once those two go together, once the ligand and the receptor molecule bind together, it allows that protein to change its shape. And often, once it changes its shape, that will then start the process of having transduction of the signal within the cell. So let's look at these receptor molecules. There are some intracellular receptors, and these are receptors that are found inside of the cell. And so they're often found in the cytoplasm, or they're found inside the nucleus. And a molecule will have to travel through the membrane into the cytoplasm, or it'll have to travel through the cytoplasm, through the nuclear envelope, and then ultimately inside the nucleus as a way of binding, recepting, triggering the transduction, and then ultimately the response. Most often, the signal molecules that are used for intracellular reception, those inside, those receptors, the molecules that can get into a cell are always going to be small, hydrophobic, and that means they can usually cross the phospholipid bilayer very, very easily. There are specific examples of intercellular receptors, and those are steroids. Steroids, more often than not, will go through the phospholipids and then bind to a receptor inside the cytoplasm of a cell. Remember, steroids are lipids, just as phospholipids are, and they're extremely small. So here's an example of what we're looking at. So here is our hormone, okay, for example, testosterone. The hormone diffuses through the phospholipid bilayer, and it comes in until it ultimately binds with a receptor protein within the cell or cytoplasm. Once the receptor and the ligand bind together, remember this is what we call our testosterone, our signals are ligand. Once they bind together, that complex then allows the transduction of a signal. And so this, for example, this hormone receptor complex would then enter the nucleus, bind to the DNA, causing mRNA and ultimately a protein to be made. And that's the response that we're looking for. So what we can talk about is those receptors that are found on the plasma membrane or in it. And there's three major types. There's G proteins, there's tyrosine kinases, and then there's ion channels. And we're going to focus on what happens is that the G-linked protein receptor is going to interact with a G protein that is inside the cell. And if you notice, this G protein gets its name because it has GDP, which sounds an awful lot like ADP or ATP. In fact, it's a very similar molecule, but this one stands for guanine versus adenine. So here's our G protein on the inside of the plasma membrane. It will interact with an enzyme. So what happens once the signal comes in is our signal will bind to our G protein receptor. Now that the ligand has bind to a receptor, the G protein on the inside of the cell will become active. And what that means is that the GDP will turn into a GTP. We have phosphorylated it. Once we have our GTP, now we have an active signal molecule, which will then go and activate our activated enzyme. Now this enzyme is active, we'll get a cellular response. As soon as the phosphate comes off of our G protein, it becomes inactive, and our receptor protein is now inactive, and the signal has stopped. Another type of receptor protein are tyrosine kinases, and these ones are pretty cool because you have to have two of them. And so if we look, we'll see that here is our tyrosine. That's why they get the name. They have tyrosine amino acids on them. And so here they are as receptors. They're individual proteins. What happens is the signal molecules must come in and bind to two of them in the signal binding area. Once they are both bound, neighboring proteins are bound, what happens is they undergo a conformational change and they hook up and bond together. We now call them a dimer, a tyrosine kinase dimer. The thing about the dimer is that it then needs to become activated. And once we have a dimer, ATP will come and phosphorylate all 6 of these tyrosines. Once all 6 of these are phosphorylated using ATP, it will now activate its relay protein, the transduction portion of the process, which will then trigger the response portion as well. Our last type of receptor that we're going to talk about are ion channels, and these are the ones we've mentioned before in normal biology. Ion channels are important because what they do is they are proteins that have a channel directly through the middle of them that allows molecules to just simply use them as a way of getting through the phospholipid bilayer. So again, let's look. Our ligand will come in and bind to an allosteric site on our ion-gated channel. Once that happens, the protein will conformationally change, creating that tunnel or tube that the molecules or ions can travel through as they then enter the cell. Once those ions are inside the cell, it then triggers its own response. As soon as the ligand is removed from our receptor, you'll notice that the gate or the conformation of the protein changes once again, and the ions are no longer free to move through.